Hi. In this presentation, I will give you an overview of clinical trial data sharing practices and specifically the anonymization practices or techniques that have been used to prepare clinical reports and data sets for this sharing to happen. I will start off by giving an overview of these practices and then talking about risk-based identification techniques, which is really the main type of data identification that I will be focusing on. So let's start off with the regulatory perspectives or what's happening on the regulatory front today. EMA Policy 70, which has been in place now for over 4 years, had 2 phases to it. The first phase required sponsors to share their clinical reports on the EMA clinical data portal once the decision has been made by the agency. Since then, there have been a number of different guidelines published by the agency to support the anonymization practices that sponsors should follow, or explaining the anonymization practices that sponsors should follow in order to prepare these documents for essentially a public release. Phase 2 of EMA Policy 70 involves the sharing of data sets, so structured data or individual level patient data, in whatever format, STTM, for example, or some other format. However, it's not clear when Phase 2 will come into effect. The EMA is in the process of moving from London to Amsterdam, and therefore the implementation of Phase 2 has been delayed, I suspect, for at least a couple more years. So really, when we talk about EMA Policy 70, we're talking about the sharing of clinical reports. And the EMA has already published more than 1 million pages of clinical reports for more than 100 dossiers on their portal. These are from various decisions that the agency has made, and these are all de-identified documents using different types of de-identification. In terms of the recommended practices by the EMA, they really follow the Article 29 Working Party recommendations. The Article 29 Working Party is the name to essentially a committee of data protection authorities in the Union, European Union, and they meet and they issue or publish opinions describing various issues around data protection and the implementation of the regulations. The Article 29 Working Party has been renamed recently, but a few years ago they published an anonymization opinion. And in that opinion, they suggested 2 general approaches for anonymizing information. The first approach requires the implementation of techniques to ensure that within data it's no longer possible to single out individuals, to link individuals, and to draw inferences. These are very strict criteria that are very difficult to meet for individual level data. So they also gave an alternative approach, was to perform a risk analysis or rate identification risk analysis, and if the results of that rate identification risk analysis demonstrate that the risk is low, then that's also deemed acceptable by the Article 29 Working Party. So the EMA essentially adopted this opinion and said you can use one of those 2 approaches, either meet those 3 criteria or perform a risk analysis. And they indicated that meeting the 3 criteria is going to be challenging in practice, and therefore they leaned quite strongly towards performing a rate identification risk analysis to demonstrate that the risk is low once the documents have been anonymized. Therefore, that's their general approach that has been used in practice to anonymize clinical reports, and I suspect it will be the approach that will be used in the future to anonymize individual level patient data as well. In the EMA guidance, they also recommended or suggested 2 approaches, a qualitative approach to de-identification and a quantitative approach to de-identification. So there is no real methodology for a qualitative approach for de-identification. And therefore, if you look at the documents that have been published by the EMA that have been anonymized or de-identified using a qualitative approach, you will find that the methodologies have varied significantly among those. Whereas for the quantitative approach, there has tended to be a little bit more consistency because there are established standards and guidelines for the quantitative approach. The other activity that is going on pertains to Health Canada. So Health Canada has published a draft guidance for implementing their regulations, which are very similar to the EMA around public sharing of clinical reports. And their draft guidance describes, among other things, how to anonymize documents for publication. Again, their focus is on documents. And they have received feedback on this guidance, and the expectation is sometime in the next few months, this will be formalized and published as a final guideline, and then Health Canada will also essentially implement a regulation that's very similar to Policy 70. So that's certainly expected in 2019, and I think it's not unfair to say that this is expected in the first half of 2019. So in general, then, there are two regulatory agencies that are now, will be soon requiring the anonymization of documents and the public release of those documents once a decision has been made. The FDA did a pilot last year, also along those lines of facilitating the sharing of documents, more limited scope. And the pilot was intended to recruit multiple sponsors to participate, but only one sponsor eventually did participate, and they have been criticized recently for having these, for not completing these pilots and not being able to bring in more sponsors to join the pilots, and the criticism was from some prominent academics. Therefore, we'll have to wait and see to what extent this criticism and this public discourse around the FDA's ability to make clinical documents and clinical reports available for public sharing, how that will play out and where this will land. But certainly there's pressure now building up for the FDA to follow suit. So we'll have to wait and see where this leads. So in order to implement these policies and regulations and to share clinical reports or structured data, for that matter, one approach and a recommended approach is to use a risk-based de-identification. So now I'll describe what this risk-based de-identification means and give you a high-level idea of how it works. So a key concept for de-identification in general is to understand the identifiability spectrum. So on the identifiability spectrum, you have at one end identified data or identified individuals. So this is data where individuals are easily identifiable. Could be data with names and addresses in it, for example. And at the other end of the spectrum is data that is considered fully de-identified, and the risk of identifying individuals is essentially zero. So these are the two ends of the spectrum. And let's start from the identified spectrum, end of the spectrum. So at that point, you have identified individuals, and then as you move down further, your identifiability is decreasing. Then you can create pseudonymous data, which has less identifiability than identified data, or original data. Pseudonymous data is data where the names and addresses and subject IDs have been either removed or encrypted. So identifiability goes down, but it's still personal information. It's still considered personal data because the identifiability is still high. And then if you continue to put in place more controls on the pseudonymous data, you get strongly pseudonymous data. So strongly pseudonymous data has less identifiability in that it's harder to identify individuals in practice for strongly pseudonymous data, but it's still considered personal information. So in most regulations, in most jurisdictions, I'd say all jurisdictions, pseudonymous data is effectively considered personal information. And then you hit this value on the spectrum This is your threshold. And once you cross this threshold, then you get to anonymous or de-identified data. So data on one side of the threshold, strongly pseudonymous, anonymous, and so on. That data is personal information. And then once you cross the threshold, that information is no longer personal, and it's anonymized, considered anonymous or de-identified information, because the identifiability has decreased so much that it's no longer considered personal information. So the objective then with clinical trial data sharing is really to anonymize or de-identify the data in a manner that is defensible, where you can demonstrate that the probability or risk of identification is below that threshold. So in order to do that, you need to be able to define a threshold, and you also need to be able to measure identifiability. So let's talk about how you can do that. A key concept then in de-identification is being able to distinguish between direct identifiers and indirect identifiers. And direct identifiers are pieces of information that can potentially directly identify individuals uniquely, and indirect identifiers are things like demographics, socioeconomic status, dates, rare events that can still identify individuals by matching that with some additional information. So examples of direct identifiers in the clinical trial context would be subject ID. And then examples of indirect identifiers in the clinical trial context would be sex, age, ethnicity, marital status, and sometimes the location and the name of the site as well. In some instances, the site ID gives away information about where the patient lives. So these are examples of direct and indirect identifiers. Now, pseudonymous data is where you have dealt with the direct identifiers. So if you remove or encrypt the direct identifiers, then you have created pseudonymous data. But as I mentioned before, pseudonymous data is still considered personal information because all the indirect identifiers are still there. They're still intact. They're still in your data. Therefore, pseudonymous data is good, and it's a precondition for de-identification, but it's insufficient. You still need to deal with the indirect identifiers. And as a matter of fact, most known re-identification attacks on data have been done on the indirect identifiers. So keeping the indirect identifiers makes it still possible to attack data sets and re-identify individuals in those data sets. Therefore, to properly de-identify data, you need to remove or encrypt your direct identifiers. So you need to pseudonymize your data, and you also need to manage the risk from your indirect identifiers. So let's talk about how you go and manage the risk from your indirect identifiers. So in general, there are at a high level, two standards for de-identification. And I'm using here the HIPAA privacy rule as an example to illustrate these two standards, because it really is one of the most prescriptive regulations globally in terms of de-identification standards. And a lot of other organizations and regulators and guidelines follow the HIPAA privacy rule. So this has two standards for de-identification. You have the safe harbor standard, which lists 18 data elements that have to be transformed in a data set. And if you perform these transformations on these 18 data elements or data element types, then the data is considered de-identified. So it's really a very simple technique, set of rules for de-identification. In general, the safe harbor is generally not considered strong enough because it ignores a lot of indirect identifiers. So as you can see in safe harbor, it really only considers two indirect identifiers, geographical references and dates. But it ignores everything else. Therefore, in many cases, you can have data sets that have a high risk of identification that still meet the safe harbor standard. Also, the safe harbor standard requires you to convert all dates to year effectively, which means a lot of the granularity in a clinical trial will be lost with respect to the order of events. So really, there are two reasons why safe harbor, as it is not an ideal standard for identifying clinical trial data. One is it's not necessarily protective enough for the very complex and detailed data in clinical trials. And the second is that you will lose a lot of very important information if you apply safe harbor. So some organizations create Safe Harbor Prime, which is a modification of Safe Harbor that tries to address some of these concerns, by allowing more date granularity. But then it's no longer Safe Harbor, and the basis for it becomes more questionable. The other standard under HIPAA is expert determination, which is essentially a statistical approach to de-identification that is consistent with risk-based methods. So these are the methods, when we talk about risk-based methods, these are the methods that we are referring to, these types of expert determination or statistical methods to de-identifying clinical trial data. There's a large body of literature on doing this. Statistical disclosure control is a discipline that has been around for 40 or 50 years. So there's a very large amount of information and literature on this topic and how to estimate the risk, measure the risk, and manage the risk. And therefore, it's one that has a large body of knowledge that can be drawn from to implement and execute. So when we talk about risk-based, we really refer to expert determination and the large body of literature that exists to support that. In practice, there are also many guidelines and standards that have been published that follow the expert determination method. So here are some from various regulators and national organizations, professional organizations, industry associations, academic institutes, etc., from various countries that describe and recommend a risk-based approach to anonymization. And there are also clinical trial-specific guidelines that have been published, again, for implementing risk-based approaches with varying levels of detail that have been published as well. And one of them, quite an influential one, is this report, Sharing Clinical Trial Data by the Institute of Medicine in the US, which has quite a detailed appendix on risk-based de-identification of clinical trial data. So there are many guidelines that essentially describe the same process, the same risk-based approach to de-identification from multiple countries. They're all consistent in principles and in methodology. The terminology varies because terminology tends to be inconsistent in this area, but the basic principles are the same around risk-based de-identification. So what is risk-based de-identification? I'll describe here the basic principles behind it, so you can understand kind of how it works in a general sense. So the first step is to set a threshold. So we saw in the spectrum chart that there is a threshold that differentiates between non-personal and personal information. So the first step is to set a threshold. What is an acceptable threshold for de-identification? The EMA has recommended a threshold of 0.09, a probability of 0.09 as a reasonable one for clinical trial data. And then after that, we measure the risk. And there are many techniques that have been developed over the years for measuring or estimating the risk of re-identification. And then you compare your measured risk to the threshold, and if you're below the threshold, then the data is considered de-identified. And if you're above the threshold, then the data is considered personal information, and therefore you have to apply transformations in order to bring the risk down and make it non-identifiable or anonymous or de-identified data. So that's the basic, these are the basic principles, the cycle around how do you de-identify data. So a key thing, of course, about the cycle is that it's quantitative. So you have to specify a risk quantitatively, and then that it is that you have to set a threshold. So the next key principle around risk-based de-identification is that of context. So context is essentially security and privacy controls that are in place to by the data recipient, and the context is important because it's another way to manage the risk. So when you're releasing data or sharing data, you can perturb the data, but you can also put in place security controls, privacy controls, and contractual controls to manage the risk. So you have multiple levers for risk management. It's not just about modifying the data. You can also strengthen security where the data is going to be processed. You can strengthen privacy practices, and you can put in place stronger contracts. So really, the overall risk is a function of the data and the context. So the same data can have a different risk in a different context. So if you take a data set and put it in a different place with a different environment, that re-identification risk for that same data set can go up or it can go down. Therefore, to ensure that the risk of re-identification is acceptably small, you have to really look at both the context and the data. The context and the data have to travel together, or they have to go together. And this is also really important. It's not just about the data, it's about the data and the context. When we talk about public data release, there are no security controls, privacy controls, or contractual controls really in place, so the context is very weak. Right? It really doesn't provide much protection. But for non-public data releases, or for private data releases, where, for example, a sponsor is sharing data with researchers, then you can put in place a context to manage the risk. Right? And what we're seeing now is more and more sponsors are sharing data beyond just meeting the regulatory requirements. They're sharing data with partners, with academics that are collaborating with companies that are doing analytics on their data, with other groups internally within the sponsor, analyst groups, etc., to do additional analysis and research on that data. And therefore, in this type of non-public data releases, the context becomes very important. So public and non-public data releases are different, and the considerations are also different between public and non-public data releases. So let's talk a little bit about how do you measure data risk. So here's an example of a data set. And so if you direct identifiers, you essentially remove all the names and telephone numbers. There's nothing there. You'd encrypt the ID. And then you look at these quasi-identifiers or indirect identifiers. So the way you measure risk is by counting the number of patients with the same attribute values. So here we have males born in 1959. There are 3 males born in 1959, and therefore we have a group of size 3. Therefore, the risk for each one of these individuals is essentially 1/3. It's 1/3. So the risk is the data risk is a function of the group sizes. So this seems relatively simple. You just get your indirect identifiers or quasi-identifiers, and you measure group sizes. Now, for data sets that are samples, it's a bit more complicated because the group sizes have to be estimated for the population, not from the sample. So this is where statistical methods come in. You use the statistical techniques to estimate group sizes in the population using the data and using priors and other population parameters. But essentially, clinical trial data are essentially samples from a larger population, and therefore you have to use statistical methods and estimators to estimate the group sizes in the population, and these are the values that you would use to measure the risk. So once you're able to measure the risk for individual patients, then you can aggregate them. For the data set, you can compute the maximum risk, the average risk. You can look at the number of individuals that are unique in the data, which means that they are in groups of size 1. So there are different ways to measure data risk once you're able to estimate the probability of re-identification of each individual patient. And then for context, like I said before, you look at the security controls, privacy controls, contractual controls, and there are various checklists and scoring schemes that have been developed to assess that. And then other factors that can be taken into account are data sensitivity, for example, and the potential harm if the data is re-identified. So another key consideration is the threshold. And we talked about the threshold. Of course, it's key. So how do you determine this threshold? Well, there are a lot of precedents for determining the threshold. People have been sharing data for a very long time, for many decades. And so a lot of precedents by reputable organizations have been set in practice. For example, here we can see the Department of Health and Human Services has used a threshold of effectively 0.5 because they only looked at uniqueness in some of their analysis. You have the CDC, which has guidelines for de-identification, and they used values or suggested values as high as 0.33. You have the NHS in Scotland suggesting some values. Centers for Medicare and Medicaid recommended a value of 0.09 for some of their data releases, which is the value that the EMA has adopted as well. And it goes all the way to 0.05. So these are all probabilities. So a lot of strong precedents. This is just a sampling of the organizations. We've done a review. There are many examples around the world. Since everybody tends to copy from everyone else, there also tends to be consistency across organizations around the world around the choice of threshold. But it tends to be in this range. So as a practical matter, if you want choose a value that is supported by strong precedents and strong practice, what we recommend is to err on the conservative side. So you take the 0.1 to 0.05 portion of the spectrum of this chart, and then we've developed scoring schemes to choose a value within that 0.1 to 0.05 range. And that is conservative, and that is based on strong precedents. And so the threshold that EMA has recommended of 0.09 is consistent with that, in that it is within that range. So the choice of threshold is not controversial because there's a lot of existing practice globally to help choose a value and base it on some pretty strong practices. And also, if you err on the conservative side, that's of course better. It's more privacy protective of patients, and that way you can have a justifiable value for setting this threshold. So essentially, then risk-based de-identification is a multi-layered approach. You essentially have a certain amount of re-identification risk, and you put in place some contractual controls to manage that risk. So that chips away at the risk, but you still have some residual risk. So you put in place some security and privacy controls to manage some of that residual risk. So these chip away at the risk, but you still have some residual risk, and that's when you perturb the data to bring that residual risk to a value below your threshold. So you have multiple levers here to manage the risk. And the advantage of this approach, of course, is that you don't have to perturb the data as much. There's always data perturbation, but you don't have to do it as much because you can manage the risk using other controls. So that way you can balance data utility and risk management and still get useful data, but that's protective of patient privacy and that meets the regulatory requirements. Now, a key thing I will also talk about here is difference between risk-based and rule-based de-identification. So we talked about risk-based de-identification and its advantages and how it works. Some organizations follow a rule-based approach, which may be based on safe harbor or they develop their own rules. Rule-based approaches are commonly used. They're very simple. However, they have some weaknesses, and therefore you should be cognizant of these weaknesses as well. First of all, when you use rules, you're essentially applying the same perturbation to all your data, irrespective of the data. So whether it's a rare disease, it's a small trial, big trial, whether it's you have 500 variables in your data set or 5,000 variables, you're essentially applying the same schema to all your trials, irrespective of the characteristics of the trials, and irrespective of therapeutic area and so on. So that's one challenge, is that it shortcuts or ignores a lot of factors. And then, of course, rule-based also doesn't take into account the context that we spoke a lot about here. It doesn't care about context because you're applying the same perturbations across the board. The other thing about rules is, and this is really important, and I cannot emphasize this enough, is that they're very hard to justify in a regulatory context. I mean, the exception is maybe the US under safe harbor, because that's the regulation, and therefore if you follow safe harbor, irrespective of whether it's a strong standard or not, it's the law, and if you follow it, you can check the box. But outside the US and other jurisdictions, or even in the US where HIPAA doesn't apply, it becomes harder to justify in that whatever identification method you use has to stand on its own. It has to be strong enough to stand on its own, to defend, and where you're able to make the argument that this is sufficiently protective. And I think rule-based approaches will be hard to justify under these conditions, especially for clinical trial data, because this type of data tends to be quite complex with a lot of information collected. So then technology and automation can be applied to automate this risk-based identification process so that it can be implemented in a scalable fashion, and a very large number of documents and data files can be de-identified at scale. And as I mentioned, as more sponsors are starting to use the clinical trial data and documents internally for their own analysis and their own collaborations, not just for transparency purposes, but to really benefit from the data themselves, being able to de-identify at scale becomes more and more important. So that's it. Thank you very much for your attention, and you can always reach me if you have any additional questions.